Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.